to Mike for inviting. And, as a new investigator, I've met a lot of people and, had a lot of potential collaborators. And uh, working with Mike has just been a distinct pleasure in that I really haven't come across anyone else who's been as supportive and who's been as productive and who's been as much fun to work with and collaborate with. So thanks so much, Mike. And it's a special sort of homecoming for me to come and give a talk here because it's where I got my start in life, but also in science. And that, the first time I did scientific experiments was some high school science fair projects just across the street at one of the Queens buildings, and did a science fair project here in Botterill Hall on E. coli as a teenager, then was a summer student here as an undergraduate. And I bring that up just to say this is where I really fell in love with science. And so for the trainees, if you find something that you like, just keep going because it's really wonderful. And then for people who are hosting trainees, you can have a huge impact on people as a supervisor. So with that, I can say that I can advance the slides. I'm co-inventor with other people at UHN on a patent, clonal hematopoiesis as a biomarker. This is unlicensed and receives no royalties. And if you're looking for sort of a state of the art on commercialization in clonal hematopoiesis, then I can save you about 50 minutes because this is pretty much the state of the art of chip and commercialization at the present time. Now, today I'll be talking about a history of hematopoiesis and clonal hematopoiesis. I'll talk a bit about my research program goals and then go through two vignettes about our work. One is on clonal hematopoiesis with the emphasis on in solid tumors, and the second is about clonal hematopoiesis and anti-tumor immunity. So research in Europe, mostly in Germany in the 19th century, so in the 1860s, 1870s, started to suggest for the first time in human history that in contrast to what was thought about the blood being produced in the liver, that perhaps there's an importance of the bone marrow for building blood, and that perhaps this is the source of blood cell production. And these ideas were highly controversial at the time and came from these painstaking experiments using the most cutting edge technologies of the day, which were different dyes that could label or at least allow for the identification of different types of blood cells. And investigators were looking into the bone marrow and now could stain for different types of cells. And what they found was that there's actually a lineage, almost a trajectory, where they could identify cells that looked like the mature cell types within the blood. So erythrocytes or red blood cells are shown here. And they could trace back what seemed to be intermediate forms of blood cells all the way back through to cells that bore no resemblance to the mature immune cells. And doing experiments that we would consider fairly rudimentary now, they showed that by doing something like bleeding a chicken and looking in its bone marrow afterwards, they saw more of these units that looked like they were related to red blood cell production. And then therefore they reasoned that perhaps these cells that bore no resemblance to the mature blood cells could be the ones producing them. Investigators went a step further by saying perhaps there's not just a blood cell progenitor or blood cell precursor that makes red blood cells and one that makes the myeloid blood cells and one that makes the lymphoid blood cells, but perhaps there is one cell or a stem cell, stemzella, that underlies all blood cell production. And again, this was a highly controversial idea at the time. It was known that this process of blood cell production that might come from this universal stem cell was also highly sensitive to perturbation. It was highly sensitive to exogenous stresses or insults, including things like radiation. In the 20th century, the atomic bombing of predominantly civilian populations in Japan indeed confirmed that radiation leads to hematopoietic failure, where thousands of individuals died of acute radiation poisoning, predominantly because they had complete cessation of all lineages of blood cell production. Strikingly, within about a decade or even less, it was shown that this process of radiation-induced hematopoietic failure could be rescued by transplantation of the bone marrow. And so in these experiments from early 1950s, this is looking at leukocyte counts or white blood cell counts in mice. Mice were lethally irradiated, and as you can see, the number of white blood cells in their circulation plummets and goes to zero. The mice in the dashed line succumbed to the radiation poisoning. They had complete cessation of blood cell production. But in these other full lines, what you can see is that mice that received a transplantation of bone marrow from an unirradiated mouse had a rescue of hematopoiesis. So they started making white blood cells again, and that allowed them to survive. And therefore, investigators reasoned that there's either a cell or some factor in the bone marrow that's being transplanted into these irradiated mice that can either rescue native hematopoiesis or can restart the process of hematopoiesis. And of course, they reasoned that perhaps this could be a stem cell that underlies all forms of blood cell production, because what I'm showing you is the graph of white blood cells, but the same graph is shown for erythrocytes and the same graph is shown for platelets that are rescued with bone marrow transplantation. And so about a decade later, there's a strong Canadian success story with the identification and isolation of hematopoietic stem cells by Till and McCullough. So Till and McCullough, in these days of after the atomic bombings, there's a lot of interest in radiation, the side effects of radiation, how to potentially protect people from it. And what they were doing was lethally irradiating mice, and then they knew that it was possible to rescue mice from this poisoning with bone marrow transplantation. What they recognized was that when they were implanting bone marrow into lethally irradiated mice, that the spleens of these mice developed lumps or these bumps, which looked a lot like colonies. And this was reminiscent of bacterial colonies streaked out on agar, which were known to be clonal units or derived from a single bacteria. And so they reasoned that perhaps these clonal units or these lumps in the spleen might have arisen from a single cell. That was important because they showed that within those lumps, they had trilineage blood cell production. So there were platelets, red blood cells, and leukocytes or white blood cells that suggested that perhaps there was a clonal unit that might be producing them. They also went further by doing things rigorously and quantitatively. And what they found was that there's a direct linear relationship between the number of bone marrow cells transplanted into these mice and the number of lumps or colonies that were formed in the spleen. And therefore, they reasoned that this one-to-one relationship may reflect a clonal unit or a hematopoietic stem cell that is sufficient to make all different forms of blood cells. Several years later, they used the sort of clonal tracking methods of the time of irradiation to genetically mark individual stem cells, and they showed that individual colonies indeed were genetic clones or clonal units. That meant that there was one cell that could produce all hematopoietic cells and maintain or restore hematopoiesis. And this led to the hierarchical model of hematopoiesis that we have today, where there's a limited pool of hematopoietic stem cells that not only self-renew or create copies of themselves, but differentiate into making all of our white blood cells, platelets, and red blood cells. And it was thought that this was a process that essentially is set after development and happens unperturbed for life without any change. Now, this developing model of hematopoiesis actually ended up being tested fairly early by a new field of genetics. And in these early studies of genetics, as you can see from the early 1960s, investigators noticed something quite striking. If you look at the age group on the left, they just use stains for chromosomes to identify within a cell how many chromosomes are present. And what they found on the right, as you can see, is that hypomodal counts, so the number of blood cells with too few chromosomes, increased with age and increased exponentially as individuals get older. And so it suggests that perhaps there are genetic changes in the blood that could be happening throughout life, and perhaps they get more common throughout life. But what these genetic changes, what these alterations in chromosome counts might mean, was completely unknown. About a decade later, Janet Rowley identified the Philadelphia chromosome. This is a reciprocal translocation that is defining in chronic myelogenous leukemia, which was shown to be disease causing. Now, this is the foundation of cancer genetics today, showing that changes in the genome of a cell can lead to a clonal disease with unrestricted proliferation. But it also teaches us that there can be genetic changes that happen spontaneously throughout life that corrupt this process of hematopoiesis. So a genetic change that might start in this hematopoietic stem cell can change its properties and lead to its transformation to cancer. But it also changes the way it actually acts in hematopoiesis. It produces too many myeloid cells, and those myeloid cells also bear the BCR-ABL translocation or the Philadelphia chromosome, and that might change their properties as well. Now, this was a process that was thought to be completely restricted to people with active cancer, to people with CML. And it wasn't until the 90s that it was identified that leukemic mutations might actually occur in normal, so-called normal hematopoietic stem cells. And evidence for this came from individuals who had been treated for acute myelogenous leukemia or AML, where a defining genetic change was detectable in their cells, even though they were in remission. So there's no evidence of cancer in these individuals. By all intents and purposes, they were in remission and potentially even cured. But when different cell populations, including monocytes, lymphoid progenitors, or even hematopoietic stem cells, were isolated, using very sensitive genetic techniques, it was possible to show that there were cells that seemed to be acting normally, producing normal hematopoiesis, but had this genetic change that was thought to be defining for cancer. Years later, hematopoietic stem cells were then isolated and functionally characterized that actually were bearing these leukemic mutations. And these leukemic mutations were called, or were believed to be defining of pre-leukemic hematopoietic stem cells. And this was thought to be a reservoir similar to polyps in the colon for the development of cancer and potentially the relapse of leukemia even after treatment. But again, these were examples that were identified in people who were sick, people who had cancer or had been treated for cancer. In another aspect or area of hematopoietic research, it was shown that these clonal expansions might actually be occurring in healthy people, and they might be occurring quite frequently. And this evidence comes from studies of non-random X chromosome inactivation. So with two X chromosomes in every cell, one of the two X chromosomes is randomly inactivated. And that means in the adult or mature population, there should be 50% of cells with the paternal X chromosome inactivated and 50% of cells with the maternal X chromosome inactivated. And what was found is that over time, with aging, there's actually significant skewing. So most older individuals will have a predominance of one X chromosome that is inactivated in the blood. This suggests that there might be a clonal advantage to one hematopoietic stem cell with either the paternal or maternal X chromosome inactivated that is overproducing or excessively contributing to the hematopoietic pool. A major advance came in about 2012 when it was shown that there were leukemia-associated mutations, including in the leukemia driver gene TET2, that were identified and enriched in people with non-random X chromosome inactivation. So elderly women who showed non-random X chromosome inactivation were significantly more likely to have mutations in these genes, suggesting that these gene mutations might actually be driving the excessive clonal expansion. And further evidence of this process of clonal expansion in individuals who are otherwise healthy with no evidence of hematological disease came from reanalysis of SNP array data that it was started to be collected for GWAS studies or genome-wide association studies in the early 2000s. So what I'm showing you here is the frequency of mosaic chromosomal alterations as detected by SNP arrays. And what you can see is that there are individuals who are cancer-free, so have no either hematological or solid tumor in the blue, or a cancer diagnosis in the red. And what you can see is that there is evidence of somatic mosaicism. So genetically abnormal blood cells disproportionately contributing to hematopoiesis becomes exponentially more common with age. And there's a signal towards people with a diagnosis of cancer having this more prominently. What was shown is that individuals who, in the blue, were cancer-free at the time of their blood draw were actually significantly more likely, shown here in the red, to develop a blood cancer. And this is fitting. If you have genetic abnormalities that might be associated with cancer in your blood, even if you don't have cancer at the time, one could reason that this is a precursor condition, that you would be at higher likelihood of developing a hematological cancer, which is shown here. Major advances were made just a few years later with cohort exome sequencing. So doing whole exome sequencing as part of large genetics association studies found that there were highly recurrent somatic mutations. So these are mutations not present in all blood cells, but only in a minority of blood cells being present and becoming exponentially more frequent with increasing age. And these mutations don't happen randomly throughout the gene or throughout the genome, but rather are enriched and particularly present in different categories of genes. And these genes are all known to be associated with hematological cancers. So some of the most frequently mutated genes are epigenetic regulators, including DNMT3A and TET2, which we'll come back to a little bit later. Whereas there are also commonly mutated genes in the regulating the DNA damage response, spliceosome, and then other things that drive hematopoietic stem cell proliferation. And this process, not just with mosaic chromosomal alterations or here looking at single nucleotide variants or indels, has been shown to be extremely common, increasingly in frequency with age, and also taking multiple forms that might be varying and may happen together or may happen separately. And so there may be mosaic chromosomal alterations or copy number changes, mutations in genes that are associated with myeloid cancers or lymphoid cancers, non-coding drivers of clonal hematopoiesis, or even evidence of clonal hematopoiesis with no identifiable driver mutation. And all of these reflect the disproportionate contribution of stem cells to the process of hematopoiesis. So if you think about having a pool of 10,000 hematopoietic stem cells or 100,000 hematopoietic stem cells, one single clone marked by a genetic mutation is doing more than its fair share of blood cell production and disproportionately contributing to the pool. But this variation and the number of different types of clonal hematopoiesis led to a need for some kind of standardization. And that led to this term of clonal hematopoiesis of indeterminate potential, or CHIP, which is something that is very frequently used, probably because it sounds easier to say than clonal hematopoiesis. And CHIP is described as a mutation that's detected in one or more, specifically myeloid cancer driver genes, having a variant allele frequency of 2% or more, and this means inferred to affect at least 4% of blood cells, and having the absence of a hematological disorder. So these are people who have no evidence of a blood problem. Now, this is a prominent example of somatic mosaicism in the blood system, but what has since been shown is that the process of somatic mosaicism is actually ubiquitous across people, so in all of us, and across all of our organs. So as we age, all of our different organs and the stem cells or progenitor cells that sustain them are accumulating somatic mutations or variants. And mutations that give that cell and its progeny a selective advantage will become more common in the population over time. Now, the blood system here is different. And the reason is that blood cells might start in the bone marrow, but they circulate throughout the body and they underlie our immune system. And so they can have diverse effects outside of the bone marrow or outside of the blood. And so, unlike other forms of somatic mosaicism, clonal hematopoiesis was found to be associated with worse overall survival. And it's known that clonal hematopoiesis is a risk factor for incident blood cancer. So as you can see here, there's a probability of developing a hematological cancer. Individuals in red with clonal hematopoiesis have nearly 12 times greater likelihood of developing a blood cancer compared to those without clonal hematopoiesis. But still, this doesn't explain why people with clonal hematopoiesis might be more likely to have worse overall survival since blood cancers remain relatively rare. And so a major advance into understanding why people with clonal hematopoiesis might have worse survival came with the definition of clonal hematopoiesis as a chronic inflammatory state, including some of the foundational work done by Mike and colleagues here at Queen's. And what was shown is that mutations in blood cells, including clonal hematopoiesis drivers like TET2, lead to monocytes and macrophages that are inherently pro-inflammatory. So they express at their baseline higher levels of pro-inflammatory cytokines. And then when stimulated, they secrete more of these bad or inflammatory cytokines than their wild type counterparts. And this was shown to create a chronic inflammatory state that is believed to be associated with a number of different underlying chronic conditions, including cardiac disease, lung disease, kidney disease. And a lot of this work has also been done by investigators, including Mike, Caitlin Flasher, Stephen Archer at Queen's, looking at correlations between having clonal hematopoiesis and having worse outcome with a number of these different chronic conditions. And yet, most individuals who have clonal hematopoiesis do not develop blood cancer. So if you look at this curve, you can see that the red bar tops out at around 5% with up to 10 plus years of follow-up. And so while clonal hematopoiesis is considered a premalignant state, the majority of people with clonal hematopoiesis or CHIP never develop a hematological malignancy. What we also know is that clonal hematopoiesis is common in patients with solid tumors. So as you can see here, the prevalence of patients who have a solid tumor with clonal hematopoiesis as well is increasing exponentially with age and is about 2 to 3 times higher than individuals who are age matched but without a cancer. And so I did some sort of back of the envelope calculations, and what I came to the conclusion of is that in Canada, there are more people diagnosed every year with a solid tumor and clonal hematopoiesis than there are with all blood cancers combined. And so as a medical oncologist, I felt that it's imperative to understand how this process of somatic mosaicism or clonal hematopoiesis is impacting on solid tumor outcomes. And so my research program goals are to determine how clonal hematopoiesis impacts the fundamental properties of cancer, including tumorigenesis and cancer risk, the biology of established tumors, as well as treatment response and toxicity. And so to this end, I'll start by telling you about our first research project on clonal hematopoiesis in solid tumors. And this is collaboration. This has really been a partnership with Mike Raw, led by an outstanding MD PhD student, Marco Buttigieg, as well as with a collaborator from Vanderbilt, Alexander Bick. And really, there's not much, or at least at the time, there was not much known about clonal hematopoiesis in solid tumors. What we knew goes back about 10 years, where there was a simple but fairly elegant study from Memorial Sloan Kettering where investigators took breast cancers and dissociated them into single cells and then used flow cytometry to isolate the CD45 positive or leukocytes, so the white blood cells that are infiltrating into tumors. And then they did whole exome sequencing or targeted sequencing to look for variants in their DNA of the cells that were infiltrating into tumors, comparing that either to buccal swabs or to cells in the peripheral blood. And what they found was that in 17 patients, 16 of them had at least some variant, so some mutation present specifically in the blood cells, or at least enriched in the blood cells that were infiltrating into tumors. And what they also noted was that many of these mutations were in clonal hematopoiesis driver genes, like in the gene TET2 here. And we essentially took this idea and integrated it with work from Mike and others showing that clonal hematopoiesis mutations can make pro-inflammatory immune cells. And we sort of combined these into a hypothesis that perhaps it's actually the infiltration of CHIP mutant leukocytes that could increase inflammation within tumors. And that furthermore, this may actually accelerate the process of tumorigenesis. If so, that could be a targetable axis for cancer prevention. And so to try to unravel this, we turn to the CP-TAC cohort, which is an extensive analysis of genetic data, including exome sequencing, RNA sequencing, as well as proteomics, from about 1,500 patients with a variety of different solid tumors. And we took advantage of this rich data set for correlative studies. First, we asked in this data set who has clonal hematopoiesis, and we used the standard CHIP definition. So we looked at the blood whole exome sequencing and found that almost 20% of individuals had evidence of CHIP mutant cells in their blood. We then said, well, let's look in the tumor exome sequencing and see if we can find evidence of the same CHIP variants, but present in the tumor exome. And what you can see is that in a number of cases, about 1/3 of the cases of CHIP also showed evidence of CHIP mutant cells within the tumor. And if that was detected, we called that CHIP tumor or CH tumor. And if it was detected only in the peripheral blood, but not in the tumor, we called that CHIP PB or peripheral blood. As I mentioned, CHIP was highly prevalent in this cohort of patients, many, actually all of whom had not been previously treated for their cancer. You can see that the prevalence varies by cancer type. So individuals with ovarian cancer have the highest rates of CHIP detected in their peripheral blood. But interestingly, there was about 1/3 of cases where in the purple, there was also detection of the CHIP mutant cells within the tumor. And that's what we called CHIP tumor. The types of driver mutations present in the blood was actually comparable to the types of driver mutations that were present in the tumor. And this was predominantly mutations in DNMT3A, TET2, and ASXL1. So we then used this idea of CHIP tumor as a correlative to see how patients with CHIP tumor fared during their cancer treatment compared to those without. And what we found is, you can see in the purple bar here, is that individuals who have CHIP mutant cells within their tumors had significantly worse overall survival compared to both patients with no evidence of clonal hematopoiesis, as well as people with clonal hematopoiesis but with the cells restricted to their peripheral blood. And while we don't know exactly why this might be happening, we looked at the likelihood that patients were free of their cancer at their point of last follow-up. And even though these were curatively intent treated patients, the likelihood that patients were tumor-free was significantly lower if they had CHIP mutant cells in their tumor at the time of diagnosis. We did some transcriptional profiling in a separate cohort and found that a signature of CHIP tumor was correlative with worse overall survival, and we used this as an attempt to try to validate our findings. And we didn't actually have to wait too long for validation of our findings, because about 6 weeks after our publication, we noted that there were some prominent publications that came out in the New England Journal describing the same phenomenon, which was known as tumor-infiltrating clonal hematopoiesis. And these other investigators found essentially the same thing. In the red bar here, what you can see is what they call tumor-infiltrating clonal hematopoiesis, so evidence of CHIP mutant cells in tumors, and those individuals having significantly worse survival compared to patients with CHIP mutant cells only in their peripheral blood or no CHIP at all. And we were able to go beyond this and try to understand how CHIP might be changing the tumors that it's present in by using the multiomics data present in the CPTAC dataset. So we compared the gene expression programs in tumors with and without CHIP mutant cells. And when we looked for evidence of different immune cell signatures, we found that CHIP tumor positive cases had significantly more immune cells, and they had immune cells that showed myeloid bias or myeloid skewing. When we looked at the gene expression programs that were highly upregulated, we saw evidence of, again, significantly more inflammation in the CHIP tumor cases. And this is looking at a pan-cancer analysis. So we wanted to dig a little bit deeper into different kinds of cancers and understand how they might be affected. And so I'm now take you through a case study of CHIP tumor in the primary brain tumor glioblastoma, which is the most common and deadly malignant brain tumor. And it's known for being a highly invasive and aggressive cancer with rich blood vessel formation. And in most cases, it's also known for not being particularly immune rich, so not having a lot of immune cells, particularly those from outside of the brain or from the bone marrow. And we want to study this cancer because when we look at the likelihood or the hazard ratio or the risk of death in people with CHIP tumor, we see that patients with glioblastoma had one of the highest hazard ratios, suggesting that they may be most prominently affected by this process. Again, when we did immune profiling from the tumors or the brain tumors of people with CHIP, what we see on the right here is, again, a strong enrichment for very high immune-rich tumors, again with myeloid myeloid skewing and a high amount of cells that seem to have pro-inflammatory properties. Because when we look at the tumors and compare the tumors of people with CHIP to the tumors of people without CHIP mutant cells within them, we see that CHIP is associated with highly uh, high enrichment of inflammatory gene expression programs like TNF alpha signaling, IL-6, and the interferon response. Not only do we see evidence of increased inflammation, but when we look at the transcriptional signatures or proteomic signatures, we see that there's evidence of strong mitogenic signaling within the tumor. So these seem to be tumors that are growing faster, dividing more, and more aggressive. We've been thinking a lot about how clonal hematopoiesis mutant cells might shape the tumor, and also wondering about whether clonal hematopoiesis might itself be shaped by the tumor. And so we've begun to look at how glioblastoma and clonal hematopoiesis may interact at the molecular level. So glioblastoma can fall into four distinct molecular subgroups, and the most aggressive is known as mesenchymal, which shows up in the green bars here. What we found is that patients with CHIP mutant cells within their tumor were significantly, nearly 8 times more likely to have this very aggressive, immune-rich form of glioblastoma. So this is defined based on a transcriptional signature. We wanted to know whether the genetic drivers of the cancer could also be different themselves. And so when we looked at the genetic drivers, so these are mutations not in the immune cells, these are mutations now in the tumor cells, we see that there are differences between the mutations in tumor cells when there's CHIP present and with a high enrichment for inactivation of the gene ATRX. This is particularly interesting because in mouse models, it's been shown that inactivation of ATRX seems to draw in more immune cells. And so we wonder whether or not this means that CHIP is related to this, as in driving a particular type of tumor, or whether this particular type of tumor is drawing in CHIP. We see the opposite effect with IDH1 mutations, where brain tumors with IDH1 mutations essentially have no CHIP mutant cells within them. And in mouse models of IDH1 mutant gliomas, we see a significant reduction in the number of immune cells present. And so this may suggest that CHIP is both influencing the aggressiveness of the cancer that arises and may also be shaped by the type of cancer that is present. It also made us wonder whether clonal hematopoiesis is simply associated with the type of cancer that arises, or whether it could actually be driving the rate of cancer formation. And so to this end, Marco began looking in the UK Biobank, where blood from nearly 500,000 individuals was banked, and these individuals were followed for up to a decade or more with health data that allows us to link the presence of clonal hematopoiesis at their time of enrollment with their health outcomes. And so we can look for the likelihood of an incident cancer diagnosis in the future based on their CHIP status in the present at the time of enrollment. And what we found was that there is a significant increase in the risk of glioblastoma diagnosis in patients who have CHIP at the time of enrollment. And this is particularly interesting because even in multivariate adjustment, we see this effect. And glioblastoma is something that is known to be associated with increasing age. It's known to be associated with male sex, but there are not strong risk factors that we understand for driving this tumor's formation. Now, we wanted to go beyond the association between having CHIP and understand whether this might be causative or correlative. And so we developed a model in my lab to model glioblastoma, and we use xenografts so we can grow human glioblastoma in mice with human hematopoiesis. To this end, we take umbilical cord blood stem cells, and we use CRISPR to either make a control mutation or induce a clonal hematopoiesis mutation, including the gene TET2. We implant these into immunodeficient mice and let human hematopoiesis begin or be reconstituting the mouse. And then once this process is taken hold, we implant glioblastoma cells directly into the mouse's brain. And what we found is that strikingly, glioblastoma growth as measured by bioluminescence was accelerated in the mice that have TET2 mutations in their blood cells. And this is suggesting to us in these preliminary experiments that perhaps clonal hematopoiesis may be driving glioblastoma growth. We can use these mice to study potential immune tumor interactions. So we see human immune cells present as evidenced in immunohistochemistry for the human hematopoietic marker CD45. And we can isolate these cells by flow cytometry, although they are very few in number. We seem to see an enrichment in immune cells in the CHIP model, which again would be in keeping with our patient data. And because we can also isolate the tumor cells, so these are brain tumor cells marked with the GFP fluorophore, we can take both the immune cells and the tumor cells, and we've performed single-cell RNA-seq. Now, we have relatively few immune cells present, as you can see in this UMAP of all of our cells present and profiled. But what we see is that even in this small data set, that there's a significant enrichment of inflammatory gene expression programs in the immune cells from mice with TET2 mutations. We correspondingly see that the glioblastoma cells from mice with CHIP are more proliferative. So they express more cell cycle related genes and seem to be more active in the cell cycle. And they also seem to be expressing more inflammatory response gene sets, including response to interferon alpha and interferon gamma. And this is suggesting to us that perhaps CHIP is actually inducing inflammation within the glioblastoma cells, may actually be driving their growth. When looking for evidence of glioma stem cell gene expression programs to perhaps reflect the most primitive state of a glioblastoma cell that's believed to be responsible for propagating the tumor, we see higher evidence of these gene expression programs in the mice with TET2 mutant hematopoiesis. And that fits with at least preliminary quantification of the frequency of glioblastoma stem cells, where you can see that there's a higher frequency of glioblastoma stem cells isolated from the xenografts of the mice with CHIP. And so to summarize part one, clonal hematopoiesis mutant cells infiltrate into solid tumors. This infiltration is broadly associated with increased inflammation and worse outcome. And the brain tumor glioblastoma is particularly affected, which may increase tumor risk. And our mouse model suggests that this may actually be driving tumor inflammation and progression. This is important because targeting inflammation may actually be a way to prevent CHIP-associated cancer risk. And evidence for this comes from the CANTOS trial. So individuals with heart disease were randomized to receive either an anti-inflammatory IL-1 inhibiting antibody or a placebo. And interestingly, there was a lower rate of solid tumors in the patients who received anti-IL-1 treatment or canakinumab, and this seemed to be particularly strong in the individuals with TET2 mutant clonal hematopoiesis, suggesting that this may be a way to identify people who would benefit most from anti-inflammatory therapies. We've also begun thinking about this and whether there could be some upsides to having CHIP in solid tumors, because in certain contexts, greater immune infiltration correlates with improved prognosis in cancer. TET2 inactivation in a mouse model was shown to potentially slow tumor growth. These kinds of mutations can activate chimeric antigen receptor T cells or cell therapy. And in our study, individuals or tumors with CH tumor, so CHIP mutant infiltration, was associated with higher expression of immunotherapy responsive gene sets. And so we hypothesized that CHIP with TET2 inactivation, so this common and pro-inflammatory genetic mutation in blood cells, may actually potentiate anti-tumor immunity. So to test this hypothesis, we developed a mouse model where we take black 6 mice that are lethally irradiated and rescue them with either a control, so TET2 wild type, or heterozygous or knockout bone marrow. And then again, after hematopoietic reconstitution, we implanted a subcutaneous tumor. So we use a colorectal cancer or melanoma line, and then treated these mice with the immunotherapy or immune checkpoint blockade, anti-PD-1, or isotype control antibody. In the mice that were treated with the isotype control antibody, we saw no differences in the proliferation of the tumor. So tumor growth was unchanged, whether or not the mice had TET2 mutant immune cells. But in the mice with TET2 mutant immune cells, when they were treated with anti-PD-1 checkpoint inhibition, we saw significantly better responses. So the tumors were smaller, and we saw greater numbers of complete responders. So evidence of the cancer essentially disappearing completely. We wanted to understand which cell types might be required for the TET2 effect. So which cell types are required to have TET2 make immunotherapy work better. And we did the same experiment except this time, in addition to using a PD-1 inhibitor, we used cell-depleting antibodies or drugs to get rid of different immune populations from the tumor. So again, with PD-1 treatment alone, the TET2 mice in red here had better response to immunotherapy. When we depleted NK cells, we again saw a better response to immunotherapy, which suggests that they're dispensable for this effect. But when we depleted either CD8 T cells and tumors grew larger, or CD4 T cells and tumors actually grew smaller, there is now no longer a difference between the wild type or heterozygous mice. And this suggests that these populations are essential for the TET2 effect. We saw the same thing when we used claudinate liposomes to deplete phagocytes. And this tells us that the TET2-CH effect requires both T cells and phagocytes. Sorry, I was going the wrong way there. We'd use single-cell RNA-seq to explore genotype-specific responses to PD-1 blockade. So from both control and anti-PD-1 treated mice, we did single-cell RNA sequencing. And with this, we looked at not just the presence of immune cells, but what we found was that the differences at baseline were not so prominent. What we saw were large differences in how the cells responded to immune checkpoint blockade. So in this plot, what you're looking at on the top is if you see something on the above the zero on the y-axis, these are cell populations that are more abundant after PD-1 treatment. And in the red bar, what you can see looking at memory, CD4 memory cells, is that they are more prominent in the TET2 mutant mice, whereas the immunosuppressive regulatory T cells were less prominent with immune checkpoint blockade. The same thing was true in the CD8 compartment. So CD8 T cells were more likely to become memory cells, and their exhaustion was much more significantly suppressed when they had a TET2 mutation. We see the same thing in myeloid cells, which were primed for anti-tumor or antigen-presenting fates with immune checkpoint blockade. And so we see more dendritic cells with TET2 mutations and significantly fewer immunosuppressive macrophages. We did some cell-cell interactions or predicted cell-cell interactions that actually predicted that there were more antigen-presenting and T-cell interactions in the TET2 mutant tumors with immune checkpoint blockade. We also looked at gene expression programs in the human tumors of patients with colorectal cancer or melanoma, as shown here, melanoma being perhaps the canonical immune therapy treated cancer. And what we see is that similar to the mice, there's evidence of CHIP-positive cancers having significantly more T-cell receptor signaling, more inflammatory and anti-tumor type immune responses like IL-12 signaling, antigen presentation, and cell-mediated cytotoxicity. And we looked to correlate the presence of TET2 mutant CHIP with patient outcome. What we saw is that in melanoma, patients with TET2 mutant CHIP were significantly more likely to derive clinical benefit from immunotherapy. And another group published that in colorectal cancer, as you can see here, a similar effect is present. So it's too small to read, but in the blue line, patients with colorectal cancer and TET2 mutant CHIP survive significantly longer after immune checkpoint blockade. And so in the last few minutes, I'll talk about digging deeper into TET2 mutant CHIP and solid tumor outcomes. And we've been trying to understand or reconcile this idea of TET2 mutant CHIP, increased immune infiltrate, more anti-tumor immunity, with our findings and other findings of worse survival for patients with solid tumors. And we hypothesized that there actually might be CHIP driver specific effects. So maybe all forms of CHIP are not necessarily equal. And in order to explore this effect on solid tumor outcomes, we needed a large data set. And so we looked at CHIP mutations identified in nearly 17,000 patients with solid tumors who were treated at Memorial Sloan Kettering in the US. And we looked at their clinical outcomes as published in this paper here. What we found as sort of a sanity check or baseline is that, as has been previously published with these same patients, individuals with CHIP are significantly older, and we saw sort of the usual suspects and frequencies for various forms of clonal hematopoiesis. And what we found strikingly is that it's actually non-TET2 mutant CHIP that is associated with worse overall survival. So this is a multivariate analysis here across nearly 17,000 cancer patients. You can see in the black line the survival for patients with no CHIP, and you can see in the gold line patients with non-TET2 mutant CHIP. And they have significantly worse overall survival, whereas individuals with TET2 mutant CHIP have no difference in their survival. And this suggested to us that perhaps there could be something good about TET2 mutant CHIP, maybe it's getting a bad rap by being lumped in with these other forms of clonal hematopoiesis. And we further broke down that gold bar of non-TET2 mutant CHIP and found that it's actually these gene mutations driving CHIP in the DNA damage response, cohesin pathway, and spliceosomes that are associated with the worst outcomes in patients with solid tumors. And this is particularly interesting and a little bit puzzling to us because these are also mutations that are associated with worse outcomes in hematological cancers and myeloid cancers. Now, most patients with solid tumors who succumb to their disease die because of cancer metastasis. And so we decided to look at whether or not a patient was diagnosed with metastatic cancer or local cancer based on their exposure to different forms of clonal hematopoiesis. And what we found, to our surprise, is that individuals with TET2 mutant clonal hematopoiesis actually had significantly lower odds of having metastatic cancer as compared to having local cancer. And this suggested to us that perhaps clonal hematopoiesis could be associated with protection from metastatic disease. We looked at the likelihood that patients with TET2 mutant CHIP have different forms of cancer metastasis. And so this is the odds that someone has bone or brain cancer metastasis. And this is, again, a pan-cancer analysis and multivariate correction. And you can see that specifically for brain and liver cancer metastases, these seem to be significantly less likely in patients with TET2 mutant CHIP. We see some cancer type specific associations for this phenomenon. So individuals with TET2 mutant CHIP with lung, colorectal, and breast cancer seem to have less likelihood of having metastatic disease. And I think most importantly, we then focused our analysis only on patients who have metastatic disease. So in patients who all have metastatic disease, if you tally up the total number of metastases, or at least allow the good people of Memorial Sloan Kettering to tally up the number of metastases, there are significantly fewer metastatic spots on average in patients with TET2 mutant CHIP. And we found this effect was most prominent in individuals with breast cancer, and individuals with breast cancer and TET2 mutant CHIP, as you can see here in multivariate analysis, also have significantly better overall survival. And so now we're trying to dig into this and understand what might be driving this improvement in survival. And so in part because I trained in the Dick lab, we're back to using these humanized mice to try to understand which cells and which signals might be driving or suppressing breast cancer metastasis. So we have a similar model where mice are implanted with either control or TET2 mutant hematopoietic stem cells. After hematopoiesis, we implant a breast cancer xenograft by tail vein injection and quantify the lung metastases that grow out. And what you can see is that with bioluminescence, the rate of tumor growth is significantly slower in the TET2 CHIP model, and the endpoint tumors are significantly smaller in the TET2 CHIP model. And so now we're really trying to dig in to connect what might be different about the TET2 mutant leukocytes and how they're interacting with breast cancer cells to either suppress their ability to grow or suppress their ability to propagate metastasis. So summary for part 2 is that patients with colorectal cancer, melanoma, and lung cancer with TET2 mutant CHIP show enhanced response to checkpoint blockade. CHIP with non-DNMT3A and non-TET2 mutant drivers is associated with worse solid tumor survival. And TET2 mutant CHIP is associated with a lower risk of metastatic cancer, and some of our preliminary data from our animal models suggests that this too might be causative. And so people have asked me, and it's a natural question, is having clonal hematopoiesis good or bad? And my answer is that it's really not that simple. And I like to use a sports analogy as a former athlete, is it good to be tall or short in sports? And I think the answer is that in most sports, it's good to be tall. And we can think about these two generational talents. So Caitlin Clark, one of the best female basketball players of all time, one of the best basketball players of all time, significantly taller than average. And in most sports, it's probably better to be tall. I can speak to that as someone who's played a number of different sports and is not tall. In contrast, you have maybe the greatest gymnast of all time, Simone Biles, who's significantly shorter than average. And so I think similarly with clonal hematopoiesis, it could be that context matters. It's the type of driver mutation, maybe even not the driver mutation, maybe it's the setting, glioblastoma versus breast cancer, the brain versus liver. And so I think there's a lot for us to uncover and unravel and to understand what is it that makes clonal hematopoiesis good? How can we leverage that to develop new cancer therapies? And what is it that makes clonal hematopoiesis bad? And how can we protect patients from those negative outcomes? And with that, I'll say thank you. Thanks for your attention. And thank you to all the people who did the work, especially Mike and Marco, and to funding sources.
Great. Questions? Go ahead.
I noticed that the T exhaustion signal was quite a bit lower in the TET2
Yes.
tumors. In the last section, I guess it was.
Yeah.
Any idea whether people are exploring, like whether that would be something worth considering with any development of, say, CAR T cells or that type of a cell therapy?
Yes. So there are some spontaneous TET2 mutations present in CAR T cells that can lead them to be more productive or effective. One of the problems is that TET2 is also a T cell lymphoma tumor suppressor. And so this and a number of other genes whose inactivation seems to make CAR T cells work better may be a little bit dangerous for producing T cell lymphomas post-CAR T. And so I think trying to find either what it is that TET2 inactivation does to make T cells more effective, or having some way to transiently inhibit TET2 during the process of CAR T may be the most effective way forward.
So just hang on a second. Charles, if you can hear me, unmute and go ahead and ask your question.
Yeah, very interesting, Rob.
Thank you.
I really enjoyed your seminar and amazing presentation.
Thanks.
I was just wondering if you have looked at mutational burden in the tumors associated with TET2 mutations, which could explain why they tend to do better. Whatever is driving clonal hematopoiesis may also be associated with increased mutational burden.
Yes.
Could you repeat that?
Yeah. So the question is whether we've looked at tumor mutation burden and in individuals with and without clonal hematopoiesis to try to understand whether or not there could be some sort of third factor that is related to both clonal hematopoiesis and better immunotherapy response or a different kind of cancer. We haven't looked specifically at tumor mutation burden, I don't think, but I mean that's a tractable hypothesis. Marco's saying no. I don't know if that's no. We've looked and there's no difference, or okay, yes. So it seems like at least so far there's no difference. And I think also probably within the mouse model at least we can isolate CHIP as one single factor and show that the effect is at least not completely dependent on tumor mutation burden.
And can I just follow up with another brief question? Yeah, inflammation is a double-edged sword. Chronic inflammation can increase, it's been shown to increase the risk of various cancers. But acute inflammation may be important for good outcomes once you've been diagnosed with cancer. So can that explain why CHIP in some cases can lead to increased risk of disease, but once you have cancer, it may actually lead to improved outcomes?
I think that's possible, but it's not something that we necessarily have tested or have data for, but it's the kind of thing that we're thinking about deeply or trying to think about deeply now is that whether there are differences in the types of cancers that form in individuals with CHIP. So is it a unique selection pressure for the evolution of a cancer? And do you potentially, in some cases, get more aggressive cancers, or in some cases, might you get less aggressive or more therapy responsive cancers? So I can't answer that question, and I hadn't considered whether it might be related to acute or chronic inflammation, but that's an interesting thought. Thanks.
Thank you.
Very nice stuff.
Thank you.
questions. The first one is, have you tried to look at post-treatment scenario? Like, for example, the DAC analysis, were there patients who you had paired specimens pre and post, and whether there was a relationship between prevalence of mutations and response?
So we haven't done that with the CP-TAC. We're looking to do that with some other data sets, including in brain tumors. But what we know is that certain forms of cancer therapy select for different forms of clonal hematopoiesis. Most of that work is focused on the bone marrow, risk of therapy-related cancers, and things like cytopenia, so problems in the peripheral blood. But I think it'll be fascinating to look at how clonal hematopoiesis might regulate treatment-related changes in the tumor microenvironment, particularly in chemotherapy-treated patients where we know there's strong selection for things like p53 mutant CHIP. And we really know nothing. I mean, we've talked about TET2 mutant CHIP. There's a little bit of a DNMT3A mutant. P53 mutant CHIP is an untapped area in cancer biology, but potentially quite relevant, especially in that scenario.
PRCA mutations.
Yes.
So do you see high prevalence, because those tumors are highly infiltrated and
Yes.
more tumor mutational burden, so especially in your breast cancer studies for ovarian, because we see, and we showed one of those graphs where it was
Yes. So there does seem to be a slightly higher rate of clonal hematopoiesis in individuals with some hereditary cancer syndromes. And I think what's also including BRCA mutations. And so that means people would be getting CHIP or certain forms of CHIP at a younger age. And I think that's reflected by PPM1D. And in fact, there's an interesting story with PPM1D that there was a Nature paper about 10 years ago saying PPM1D is a new hereditary cancer gene for ovarian cancer. But what it turned out to be was somatic mutations in blood cells that were either related to the likelihood of getting ovarian cancer, or they were selected for post-platinum. So, I mean, the paper stands, but the interpretation is a little bit different in light of what we know about clonal hematopoiesis.
Yeah.
No, we've just started trying to do some spatial analysis in liver cancer, but we don't know yet. So hopefully we'll be able to share some data with you in hopefully not too many years, but not yet. Thanks.
David?
Yeah.
Maybe a very naive question. Is it clear that CHIP is having an influence on the microenvironment or on the tumor cells?
Yes.
Or does it depend on the context
Mhm.
of the tumor type and the type of CHIP?
Yeah, I think that's a very astute point that we talk about the microenvironment and imagine that this is a relationship between the CHIP mutant immune cell and the tumor cell in direct proximity. I mean, we know from work from Mike and others that people with CHIP have higher levels just in the serum of circulating cytokines, and so that could be having a role. There may be relay cells in between the immune cell and the tumor cell that could be transducing some of these signals, or it could be those cells that are even more potently affected by the CHIP mutation. So in glioblastoma, we also see increased angiogenesis signatures, and whether that could be the factor that's most important for changing tumor biology, we haven't yet explored. Thanks. Yeah.
About your slide on the ICV mouse modeling.
Yes.
I think I noticed that you did a heterogeneous knockout.
Yeah.
As well as full knockout.
Yes.
And there was only an effect seen with the head mice.
So
Not the full knockout.
yeah, we saw the same effect I guess we had fewer mice with the full knockout, and so it was not statistically significant. It had a p-value just above 0.05. So we saw the same magnitude and change, but it was not significant in the full knockout. And then in the paper, we say, most people with TET2 mutant CHIP have just one allele inactivated, and so we're going to use the TET2 heterozygous mice. the reality, maybe should not record this, is that we just couldn't get more of the tattoo knockout mice. And so we were kind of forced to use the heterozygous mice, but still saw the effect. And so in the end, it worked out in that we had this justification for using the different model. Or the more relevant model.
You still have the advantage that you have a lot of hematopoietic cells that have
Yes. And I will say that, you know, in our model, when we really started to titrate down and use less tattoo mutant bone marrow cells, we didn't see the effect. And so it seemed to be dependent on having a lot of tattoo mutant immune cells.
So this may get something we were talking about earlier.
Yeah.
In the mouse model, you're trying to elicit this difference.
Yes.
In that model in the course of four weeks, maybe.
Yes.
Less, where it might take a lot longer.
Yes, and that we were also speaking today about how we've been doing this in subcutaneous tumors, but really want to start doing this in spontaneous tumors where the tumor from its very initiation or origin grows in the context of chip or no chip. And so those are experiments that take longer but are starting.
If there, oh, you have another question, please?
Yeah.
Maybe on that,
Yeah.
talking about the tumor microenvironment interactions with different cell types. As I guess the main message was about chip influencing anti-tumor immunity via T cells and phagocytes. So then, do you think those T cells and phagocytes are chip mutant or wild type?
So in our model, the T cells were mostly TET2 mutant, and the myeloid cells were essentially all TET2 mutant. Now, in the second paper, this other paper that was published from MD Anderson, they actually found that the T cells that were infiltrating into their pancreas cancer model were not TET2 mutant. And so these were interactions between myeloid cells that were mutant and T cells that were wild type. And we're working to understand whether there are actually TET2 mutant T cells in human tumors. And I think that there are, and I'm hoping that we can find them and show people.
No other hands in the room and none on the Zoom. Join me in thanking.
Thank you.
For wonderful time.